Thank you for an opportunity to present to MESARC Showcase on genetics to improve hybrid and Eurasian water milfoil management. Um, I'll acknowledge my collaborators now. Actually, probably the lead collaborator is Ryan Thum, who's associate professor at plant sciences at Montana State University. And then two of his grad students who we've been working with and publishing some of our results, Katie Gannon and Hannah Hoff. So this presentation will be, if you've seen me talk before, some of this will be review. New folks, then this will be an introduction to the topic, but so bear with me if you've heard some of this before. But invasive Eurasian water milfoil was introduced in North America from Eurasia in the 1940s. It showed up in Minnesota, well, at least it was first documented in Minnesota in 1987. Just mention this is there were probably several introductions to North America in the 40s, early 50s, but it wasn't a widespread set of introductions. But it started spreading since that time. It's now present in 48 states, and it's probably one of the most widely managed aquatic weeds, certainly in North America and also globally. It forms these extensive mats that you can see here on Lake of the Owls, and also my screen behind me that inhibits recreation. It's difficult to boat through. It shades the bottom, outcompetes a lot of native plants, ends up with a monoculture, which isn't great fish habitat. It's present in probably close to 400 lakes now in Minnesota. This is a map with the distribution of Eurasian water milfoil, and those black dots across the state. This is from 2017. Now there's a very close relative, northern water milfoil. So Eurasian water milfoil is Myriaphyllum spicatum, and northern water milfoil is Myriaphyllum sibericum. And actually back in the 50s, these are considered, sibericum was considered a subspecies of Eurasian. So they're closely related. But the native northern water milfoil is very widespread in Minnesota. And prior to Eurasian water milfoil being introduced, it was one of the most permitted plants, so for control. So people were still controlling nuisance populations of northern water milfoil. In deeper water, it can be outcompeted by Eurasian water milfoil or in poor clarity conditions. So northern seems to do a little better in good water clarity and or shallow waters. The northern and Eurasian are closely related, so they can hybridize. These both produce vegetatively or clonally, so by breaking off and fragmenting, but they also reproduce sexually. So when they reproduce sexually, then they can interbreed and producing a hybrid of Myriaphyllum spicatum and Myriaphyllum sibericum. These hybrids also reproduce sexually and vegetatively. And the work that Ryan's been doing is sort of indicating more that there's a lot of reproduction probably going on with the hybrids, and perhaps more so than there is within the pure strain Eurasian water milfoil. It's very difficult to distinguish water milfoils. One of the simplest ways and actually probably most effective way is to do leaflet counts. Eurasian water milfoil tend to have a large number of leaflet pairs, 12 to 20 leaflet pairs. Northern water milfoil tends to have much fewer leaflet pairs, 5 to 11 leaflet pairs. And hybrid water milfoil ends up being intermediate. But the problem is where you can see northern and Eurasian don't have really much overlap, although again, there can sometimes be a question with that. Hybrid is intermediate, and it's very difficult if you're in the middle there to determine whether you've got Eurasian water milfoil, northern water milfoil, or truly a hybrid water milfoil. This is just another plot sort of giving you an idea that you can also look at leaf length in addition to pinnae. So these pinnae, this is going to be double those leaflet pairs. So 30 pairs, it's going to be, 30 pinnae is going to be 15 pairs, but you can see that up here in blue, Myriaphyllum spicatum, down here in green, our native northern water milfoil and hybrid in between, but a lot of overlap. So it's really difficult without genetic analysis to determine what you've got. So genetic analysis is really essential to actually distinguish these. Morphology is not sufficient. Hybrid water milfoil It does appear to be fairly widespread, especially across the Midwest, but it's also present in New England states and present out in Oregon, Idaho, Washington. It was first documented in Minnesota in the late 1990s. And so actually some of our hybrid water milfoil as part of Michael Moody's original work on this. And most of the work has been done in the upper Midwest. So most surveys for that. So for example, this is somewhat old now, but Wisconsin had identified more than 144 lakes with hybrid water milfoil populations. So why worry about hybrid water milfoil? Well, just because it's a hybrid, it could be more vigorous or invasive. So have faster growth and be a better competitor. What most of us are concerned about is it could be resistant to or tolerant of herbicides or biological control agents. So far, there doesn't seem to be great evidence of resistance to biocontrol agents. Hybrid is sort of intermediate between Eurasian and Northern. But there is evidence of increased tolerance to herbicides in some populations. And this has been well documented in several places in Michigan and elsewhere of tolerance to 2,4-D and triclopyr and also to Florida. So our management could be selecting for resistant or tolerant genotypes, and this could end up resulting in being a lot more difficult to control Eurasian water milfoil if we keep selecting for these resistant genotypes. So again, this is our current distribution of Eurasian water milfoil, almost 400 water bodies. I'll just mention that the DNR again, because it's difficult or impossible to determine what you have for sure without genetic analysis, and considers all of the water milfoil to be invasive water milfoil. So Eurasian and hybrid are both lumped in together. They've got some Eurasian component to it. So those will be considered introduced or sort of invasive water milfoil. So the DNR database doesn't distinguish those, mainly because most of their records are based on visual IDs. So now I'm going to give you some updated information. Again, we've presented this in previous talks, but in 2021, Ryan ended up adding information from 17 additional lakes sampled in Minnesota. So now we've assessed a total of 93 lakes for genetics. 65 lakes have Eurasian water milfoil, 44 have hybrid water milfoil, and 33 have northern water milfoil. It's a general tendency for the northern water milfoil to be more prevalent outstate, hybrid water milfoil to be more prevalent in the metro, and Eurasian water milfoil seems to be widespread. I'll just point out the original introductions to North America were of pure Eurasian water milfoil. Again, there's evidence there were probably only a couple genotypes, 2, maybe 3, that were introduced. And then for those to end up diversifying, they would need to back cross or start reproducing. So for our work, we wanted to do hybrid genotyping to determine what the diversity of the milfoil is present, if there's differences in diversity among these tax or geographic settings, and identify potentially problematic genotypes. So to look at the genotypes now, so this is no longer just looking at whether it's hybrid or Eurasian. Eurasian water milfoil, we've now identified 16 genotypes. One of them is very widespread. So it's present in 62 lakes. That's the dominant genotype in Minnesota. That was probably almost certainly the one that was introduced back in the late 1980s, and it is spread clonally throughout the state. However, 7 lakes did have more than one genotype. And I'll just mention this is a bit of an update from the work previous to, sorry about that, previous to 2021, when I think we only had like 8 or 10 Eurasian water milfoil genotypes. So we have detected a little bit more diversity in Eurasian water milfoil than we had previously. Michigan has more diversity of Eurasian, pure Eurasian, than we do yet in Minnesota. 7 lakes did have more than one genotype of Eurasian water milfoil. Northern water milfoil is very diverse. There's over 96 genotypes that we've identified, and so far we've not found a genotype in more than one lake. So Each lake has a different genotype, and many lakes have multiple genotypes, so a lot of reproduction going on with the northern water milfoil. Hybrid water milfoil, we also have 96 genotypes now that we identified. So again, this is an addition from what we had back in 2020. Generally, most lakes only have one unique genotype of hybrid. They may or may not also have Eurasian water milfoil. But 17 lakes did have multiple genotypes, and one lake, aside from Lake Minnetonka, had 6. Lake Minnetonka is quite diverse. We've got those diverse bays there, and there's actually a total of 24 hybrid water milfoil genotypes in Minnetonka, plus the one predominant Eurasian water milfoil genotype. And in each of those bays, Grays Bay, Phelps Bay, so on, there's 5 or more genotypes present, plus Eurasian water milfoil. Now, there are 8 genotypes of hybrid that we found in multiple lakes. So this has to be clonal spread. So if they're identical, then that's going to be just spreading a fragment from one lake to another. They're not reproducing sexually in those lakes. One genotype has now been found in 12 lakes. So that's an expansion from previous, and 7 genotypes have been found in 2 or more lakes. So this just giving you an idea of this. Green is this widespread Eurasian water milfoil genotype sort of around spread throughout the state. Doesn't seem to be any clear pattern in the number of genotypes that are present there. And so this is all clonal spread that we've had either by boaters, birds, or drift downstream. Hybrid water milfoil is more common in the metro and nearby counties. And we do have some of these genotypes. You can see, for example, this red dot here that ends up being shared among a number of lakes. So this genotype has now been shared among 12 lakes. Other than that, there's no clear pattern in genotype distribution that we're able to tell. So this one widespread genotype is present across these 12 lakes. And again, these are not all connected by waterways. So again, it's probably boater connection. You can also notice Lake Charlotte out in Wright County is quite distant, and a couple of these lakes up here. So could be boater traffic, could be someone suggested some bird movement patterns, but shifting that genotype around. So here, just whoops. We'll just mention here that, right, there is this clonal spread then of some hybrid water milfoil genotypes that's going on. And then in these other cases, these genotypes have probably developed within that lake or in a nearby lake, and we haven't identified another source yet of the hybrid. So again, most lakes only have one hybrid water milfoil genotype, but there are some areas, and particularly like Lake Minnetonka, which is a quite diverse set of habitats, and it's also had Eurasian water milfoil the longest, since 1987, to end up diversifying. So it's got a lot of diversity in hybrid water milfoil genotypes. Interestingly, it doesn't have diversity in Eurasian water milfoil genotypes. So further update on this. Brian's been analyzing those additional samples from 2021, and he also, people have been sending them, including our lab, samples from 2022, which haven't been processed yet with some Army Corps of Engineering funding that he has. Currently, all of the work has been done by microsatellites, and he's hoping to shift the genotype by sequence, but that's progress has been slow, but I think probably by next year that they'll be moving into genotyping by sequence. And that will be a little better, because once we end up doing that, actually the analysis will be less complicated. You'll be able to directly identify what a genotype is. And then furthermore, you may be able to look at specific regions and determine traits that are related to resistance or perhaps growth characteristics. So you'd actually be able to home in on those components to look when you look across the genotypes. So we do now have updated genotypes. The data I presented today includes the 2021 data. And we've also developed a story map for genotype distribution. So I'm just going to take a minute here to share that. See if I can pull that off. And if I can figure out. I'm going to share that. So in a previous map that we have just shows whether you've got Eurasian water milfoil, northern water milfoil, hybrid water milfoil. But now with the help of McDurr and also use spatial, we've got a storybook map that ends up showing distribution of genotypes. So both of hybrid water milfoil, Eurasian water milfoil, and northern water milfoil genotypes. So you can zoom in. This is our map and distribution. This is still with data up through 2020. So we haven't added the 2021 data yet, but we'll be doing that in the next few weeks or month. And now you can see the lakes that have Eurasian water milfoil. Also, the lakes that are outlined in blue here are lakes that the Minnesota DNR has listed as infested, but we haven't sampled those, so we don't have records. So it's only the lakes with the circles or the ones that we have genotype information. But we can look here now, for example, at Crooked Lake. It's got 3 different genotypes of hybrid water milfoil. Some other lake, let's look at Spectacle Lake, for example, has Eurasian water milfoil and 4 genotypes of northern, but no hybrid water milfoil. So now you can look at, for example, your particular lake and determine which genotype you might have. And then you can also look and see if that genotype is present in some other lakes. So let's see, for example, we could try to go on here and look at Otter Lake. We've done a lot of work there, and there's this hybrid 03. So we can actually grab that hybrid 03 and think about it. We can move here and end up trying to do a search then on a genotype. So now we can click apply and find out which lakes have that hybrid 03. Now I happen to know that hybrid 03 in this case is one of the widespread genotypes, hybrid genotypes. So you can see it's listed in a number of lakes here, including this lake out in Lake Charlotte out in Wright County. So we can look at the different distributions of genotypes. So we think this will eventually be quite important. And then we also hope also to start trying to link some of this information to management information. But this is sort of where we are at the moment. It's going to allow you to determine if you have a particular genotype within your lake, and then what other lakes may have that genotype. And then if we end up identifying, say, for example, I don't know, genotype 149 is resistant to an herbicide, then that would be something we'd want to look for. And you might want to check and see if that's in a nearby lake. So let's see here. I'll go back to my PowerPoint if I can do this. Okay, so I'm hoping I'm back on the PowerPoint sharing. So for distribution, our hybrid water milfoil is widespread, but it is more common in the metro. We haven't done as much sampling out state, but again, hybrid Eurasian water milfoil hasn't been up in some of those sites as long, so that's probably one factor. Northern does tend to be more common outstate, and that's partly because it's probably been less impacted by water clarity and also by management activities in the metro. Eurasian water milfoil does have very limited diversity, and northern is highly diverse, and Eurasian and hybrid water milfoil ends up being sort of intermediate. One thing interesting is the parent taxa do not need to be present to occur, but it does appear like most hybrids are from in situ reproduction. So at some point there probably was Eurasian and hybrid, or Eurasian and northern present in the lake, and then they ended up reproducing, and then the other those parent genotypes disappeared or became rare, and we were not able to find them. And there is human-mediated dispersal. So we've got genotypes that have been distributed around the state by human transport. So that's sort of our study of distribution, and we're keeping an eye out for that, looking for these changes and broader patterns as we keep getting more information across the state. But the next thing we're doing is looking at response to management. So one of the intensive things we did on our fairly recent NASERC project was looking at treated and reference lakes in the metro with point intercept surveys, and then characterizing the genotypes present in those lakes before and after treatment. So Katie Gannon analyzed these data as part of her master's thesis, and we have a paper that just came out this year in Aquatic Plant Management on that work. So these are the lakes that we ended up looking at intensively. There are a few other lakes that we didn't end up including in this published study, and you can see that we've got an array of herbicides, 2,4-D, Procelicor, and Fluoridone are the ones that we followed in this study. So for responses, just give you a couple examples. This is North Arm and Lake Minnetonka, pre and post treatment, and you can see pre-treatment there's a lot of Eurasian water milfoil around. Post-treatment, Fluoridone essentially eliminated all. There's one stem of Eurasian water milfoil found. Similarly in Schmidt Lake, again for several years after treatment, after Fluoridone treatment, Eurasian water milfoil was not found. So far in Minnesota we have no evidence of resistant genotypes to Fluoridone in Minnesota. There is at least one, and I think several genotypes that have been identified, and one widespread one in Michigan that is causing lots of problems there. So we'll hope that doesn't show up in Minnesota. But so far, no resistance in Minnesota to Fluoridone. This is some response here to Procelicor. So we're looking at Grays Bay and Ham Lake. And you can see in general, those treatments are pretty effective, although again, in some cases, we still have plants left after that. So again, we're sort of continuing to monitor those plants after those treatments. And it gets hard to separate sort of ineffective treatments or these spot treatments that may end up not having a widespread impact versus potential resistance. But we can use this to sort of determine if we've got some candidates that are really likely to be resistant. And then this is just an example of responses in bald eagle. Again, you can see that we're fairly effective in reducing the Eurasian water milfoil. And again, the pure Eurasian water milfoil here, E1, tends to be fairly susceptible, it seems, to herbicides we use. And actually in bald eagle, we ended up getting a bit more northern water milfoil recovering in those areas. And that's unclear whether that's because we didn't treat those areas as much and it just recovered, so selective targeting of treatment plots, or whether there is some resistance there. One of the classic things that got us interested in this was in North Arm, and this is some treatment with triclopyr. And when it was treated in 2015, again, you can see that there was a distribution of hybrid genotypes here, a number of them, I think about 8. And in this case, 2 different genotypes of Eurasian that were present. And then after treatment, most of those the Eurasian genotypes disappeared. Hybrid genotypes were all reduced except for this one, H8. So it became, and even in some treatment plots, became dominant. And so that was sort of a candidate for a potential concern that we'll look at. Let's see. Yeah. Now, if we end up looking at Fluoridone again, so that strain was present in North Arm with Fluoridone. So that's this MCH7. It did seem to be fairly well controlled by Fluoridone, although it has recovered back by 2020, has ended up coming back. But it was quite well controlled in 2018 and certainly suppressed, only found in a couple of sites in 2019. I think I'm going to skip through. These are sort of just looking at response of these different strains, again, across time to management. I'll just wrap up this sort of field assessment of this with an interesting observation from Phelps Bay here, where it was treated with diquat and Procelicor. Procelicor is a lot easier to say than this name here, so I will just stick with Procelicor. But you can see that after treatment in these areas, the remaining strain is a northern water milfoil strain. And so we've now collected some samples. We're going to run that into our test. That's sort of a benefit now if we've got northern water milfoil that may end up being resistant to some herbicide treatments. So we had good control of the various hybrids that were present there with Procelicor and diquat, but we end up having some northern water milfoil. Now, this could also be a source of resistance, right? If it hybridizes, this northern water milfoil hybridized with Eurasian, that trait was carried through, then we could end up developing a resistant strain. Okay, so for management, we've got sort of variation in response to the different taxa and genotypes to treatments. Spot treatments and adaptive management are hard to assess. So we've got these smaller treatment areas. It is really hard to assess whether those changes are because we treated that area or because they were on fringe areas and then expanded back into those treatment plots. But we have been able to identify some genotypes worthy of further investigation, including one northern and several hybrid genotypes. And this is going to guide additional testing of specific genotypes for response to herbicides. So that's where we come into this utility of genetic monitoring. And Hannah Hoff sort of used that then as a springboard. Katie sort of set up the assessment of that, and Hannah to tie this in to see if we can, once we identify those genotypes worthy of investigation, then to see what their response is to specific herbicides. So again, back to North Arm, I already showed you those data. I apologize, we keep changing the names of these genotypes. I think we're coming up with a consistent convention moving forward here fairly soon. You can see this Min1 strain, which was H7 or H8 in the previous work, again, responding in North Arm. So it ends up persisting after, after triclopyr treatment. But this also happened in St. Albans. So this is the other reason why we thought that this is a good candidate for further questions. So we come in St. Albans Bay, there was only present a 13% occurrence, and we had a wide range of other genotypes present, both Eurasian and northern watermilfoil. And then after herbicide treatment, there was only 100% occurrence of not very many sites, but of this Min1 strain. So this hybrid is a candidate for further study in response to oxymimic herbicides. And then we ended up testing it with challenge tests. So if you really want to know if something is resistant or more tolerant to an herbicide, or if it's more aggressive, you really need to bring it in and come up with some common garden laboratory experiments to determine this. So what we're doing is exposing known genotypes to controlled doses of herbicides and common garden experiments with suitable controls. So in our lab, we're currently assessing 5 genotypes, the widespread across the state Eurasian watermilfoil that we collected from Christmas Lake. There's a widespread hybrid that is from Otter Lake and also Olson. It's found in 12 lakes. And then a hybrid from Ham, a hybrid from North Arm, and a hybrid from Smiths Bay. Now, notice that there are several different hybrids in North Arm and Smiths Bay. Okay, so we know specific ones that we're actually dealing with. So we can plant these into cups and put them into the containment lab. If you do the tour tomorrow, you'll be able to see we've got some experiments going on now similar to this. We end up establishing the plants, and then we can end up treating them with the herbicide, follow that through time, and see what happens. So right after 72 hours exposure, we change the water. So again, 72-hour exposure with that dose, and then allow them to grow for 6 weeks and sample them. So this is the result of the 2,4-D experiment we had. So you can see here that a couple of these blue genotypes. Well, let's go to the light ones, Christmas at the yellow line, and it's light blue, Ham, and Otter, all end up being reduced pretty significantly in the treatment from where they were in the control. But if we look at both Smiths Bay and North Arm, there's much less response, much less reduction, suggesting that these are less affected. And in fact, if we end up doing a comparison of treatment and controls there isn't a difference between North Arm and treatment and control, and Smiths Bay and and treatment and control, but there is between for Ham, Otter, and Christmas Lake. Now, Ryan's lab, we sent the North Arm material to him, and then also some of our Christmas, and then he has another population from Montana, or not from Montana, but from Michigan that he's keeping in Montana that's the same as our widespread population here in Minnesota. And he ran dose response tests now. So this is, again, with 2,4-D at 0 micrograms, 500, 1,000, 1,500, and 2,000 micrograms per liter, and looked at the response of these. So again, we're getting a dose response curve on biomass. So this is control, high biomass, and then it declines as we end up increasing our treatment. Well, you can see that our North Arm genotype here has much less reduction, and actually these end up not being significant out to 500 or 0.5 milligrams per liter, and then at higher doses it does end up decreasing. All of the other clones in this case, or genotypes, do respond pretty directly to the treatment. So again, now this indicates that North Arm does show resistance to 2,4-D, especially at doses at 500 micrograms per liter or less. So from our challenge test, we've got evidence that North Arm hybrid and Smith's Bay genotypes are resistant or tolerant at 2,4-D. And North Arm results were confirmed with Hannah's gradient test out in Montana. We're now running gradient tests, and we're actually decided to start looking at triclopyr. So we're continuing those tests. And we'll continue these challenge tests to test these genotypes with different herbicides. And then once we do that, we'll move in at some other candidate genotypes with GLRI funding. So as I mentioned, we're currently testing triclopyr, and we've got some evidence that there is at least one and probably two genotypes that may show some resistance to triclopyr. And it isn't necessarily the same ones that we're dealing with 2,4-D. And again, originally the thinking was maybe oxymimics, they would all respond the same, but we have some suggestion that there may be some difference between triclopyr and 2,4-D responses. So in summary, hybrid watermilfoil is widespread across the state. It's reproducing. There's a lot of genetic diversity in hybrid watermilfoil. So this is where we've got this opportunity for either more invasive and aggressive genotypes to develop, or ones that are resistant to herbicides. So we have evidence tolerance to herbicides and possible invasiveness for some genotypes. And again, it's genotype specific, not just hybrids in general. And additional testing of these genotypes for response to herbicides is ongoing. And we hope to be able to develop a catalog so that eventually people will be able to look and say, oh, if I've got a genotype X in my lake, I probably should not use herbicide Z. I should use herbicide Y or something. Again, there's more information on this on the MACERC site at hybrid distribution. And with that, I'll just acknowledge our funding, original funding for several years from the Environment and Natural Resources Trust Fund, LCCMR. Current work is being funded by the U.S. Fish and Wildlife Service Aquatic Invasive Species Interjurisdictional Grants for the Great Lakes states and tribes, and that's going through Montana State. And then we really thank cooperation from the DNR, AIS specialists, and applicators such as Patrick Seltzer. Where am I going here? For a lot of help. And James Johnson also is greatly appreciated. And then Jasmine Altawilli was key in collecting a lot of early distribution data. Jane Feely and John Jarrettson helped out with those lab studies, and Greg Turok at Montana State. So with that, I'll see if anyone has any questions.
Thank you, Ray. All right, we have a few questions that have come into the Q&A box. Just a reminder to drop those questions in there. We'll start rolling through them. Again, just click on Q&A in the bottom of the window. The first question that we have is, because northern watermilfoil is much more diverse in the region than Eurasian watermilfoil is the high diversity of hybrid watermilfoil primarily derived from variation in hybrid watermilfoil ancestors? That's in the box too, if you want to read it, Ray. That's helpful.
Yeah, and I probably should think about that a little more, but I think the way we're defining genetic diversity right at the moment really is just whether things are sexually reproducing. So with these, if they reproduce at all, you're going to end up coming up with a different genotype that we can identify. So originally we're looking at this as clonal reproduction. They're essentially the same, except for potential for some somatic mutations that would occur here. In that case, probably the bigger key really is that diversity is sort of coming from this propensity to reproduce. Now, as far as some additional traits that go in, it is more likely, since you've got a whole bunch of different northern watermilfoil, then you're more likely to find one of those that ends up being, say, more tolerant to a particular herbicide or expressing a certain growth factor. And then things that we don't know is there probably could be a lot of interaction there. So it could end up being, having a allele from Eurasian and Northern combination could end up providing an important response.
Thank you. Next question is, if a lake group or county group want to get a sample analyzed to determine if their lake has hybrid watermilfoil, what is the process?
Right. So probably, I mean, has been some grant money. I could throw this a bit to Jake at the DNR for consulting with him. You can also get a hold of me or Ryan, and we can think about it. Again, there's two levels of trying to think about this. The first is just, do you have hybrid watermilfoil versus Eurasian watermilfoil? We tend to think that this isn't a real interesting question because unless you know specifically what you have, just knowing that you have hybrid versus Eurasian doesn't give you a lot of information. At the moment, it's a little cheaper and faster to test to determine that. But then if you want to know what genotype you have, you got to go through the process again. If we're looking for specific genotypes and we don't have information from a particular lake, then it would be nice to get a range of samples. So just going out and getting one or two plants isn't probably as good to sort of, see if you've got some diversity in your lake and getting more than a couple samples to make sure that you end up trying to capture some of the diversity there. So I'm not sure whether Ryan next year is going to have enough funding to end up being able to cover this. This year, he was able to cover a set of samples. So we can check in with Jake Walsh at the DNR and then Ryan and see what we'd do moving forward.
Okay, so it sounds like you'd need to determine that genotype of the hybrid. And to do that, it would be sending in those samples. Is that
right?
Correct?
Yeah, and even if we are just determining whether it's hybrid or Eurasian, we'd still need to do a genetic analysis. It's just sort of a different genetic analysis. So the one sort of using ITS is sort of a rough and dirty, fairly quick way to go. And to actually genotype it, it gets a bit more complex because you need to compare it to everything else.
So this is the last question on the list. I would encourage anybody in the audience, if you have additional ones, pop them in there and we'll move on to them after this one. Otherwise, we'll wrap up a little bit early for lunch. But Ray, the map that you shared, the interactive map that you and Macer staff put together,
Right.
Is that the best place to go and check for your lake or your area to start gathering that information? Or are there other resources that people should be tapped into?
Well, that would be the best place now if you want to determine which genotype is present in your lake. I'll just mention at the moment, the numbering system, as you saw, will end up being flexible. And probably when we put it in, I'll put in one new set of numbers. And then we're hoping probably next year, Ryan will have a consistent labeling thing. So genotype X is going to stay X for the rest of its life rather than changing numbers when he redoes a set of analyses. But so, and if you just want to know what is present in your lake, then we do have that other map on distribution of whether it's Northern Eurasian or hybrid, and we'll also update that at the same time. But I think the story map moving forward, if you want to know your genotype information, I do see that if you want some additional, and we do have other information about hybrid watermilfoil on the Macer website on that one page, and then John also pasted information about the genetic identifications from Ryan's lab that if you're interested in more analysis.